so we left the best for last. yeah, of course. It's a great honor to welcome my friend Jorge Velasco-Hernández. He also requires absolutely no introduction whatsoever. So I'm just going to say, Jorge.
Yeah, thank you very much, Anna. Thank you for the invitation to talk in this workshop. I hope it's interesting to you, the topic. And I chose it because we were discussing about reproductive numbers and the role of the reproductive numbers and the role as thresholds for indicating certain type of behaviors or changes of behavior in the dynamical systems that they describe. And I thought this was probably is also interesting because it illustrates another kind of complexity in the study of epidemics. So I come from Mexico, the Institute of Mathematics. I am a biologist by training. And I've been involved in this kind of things some time. So let me see how it goes. What I'm going to do is to illustrate that in order to have an epidemic, we need to have an infection at the individual level. If you have enough viral loads at the individual level, then you may be able to transmit the disease and you have an outbreak, at least. The study of what they call immunological or within host dynamics is long, particularly grew up very strongly with the AIDS problem. Alan Perelson developed several models for that. And there are many, many ways of modeling immunological dynamics. It depends on if you consider memory in the immunological system and many, many things. So I'm going to use very simple description, but it's still a classical description of these models. To illustrate the idea, probably this is too small, but essentially the main components that I'm going to consider, this is for COVID, so not because I'm going to talk any special about COVID, but because I think we have it all in our minds. So you have a virus, it penetrates the body, then it goes into the cell. You can have different treatments in different stages. Then the protein coat dissolves, and then all the genetic information goes into the nucleus. Then the nucleus codes the new virus and the new infection, and they are released. So you have the infection of target cells. These target cells serve as factories to reproduce the virus. And therefore, this damages the body. This is the disease. And because the virus essentially either bots from the several mechanisms, but essentially either is bodying mechanism or it explodes the cell and liberates. And then it causes death to the cells. So this is the basic structure of the immunological process. The epidemiological process, you are very familiar with it. We've been discussing particularly one type of diseases, which are SIR. Before this talk is SIS, and I think Tom talked about SI. But this is only a very small subset of the possibilities that exist in nature for infectious diseases. Essentially, we are been looking in this workshop to human transmission, using as examples essentially COVID and some abstract constructs. But essentially, there are many, many more diseases. In fact, many of the more important diseases, the one that causes more mortality, are not directly transmitted diseases, but vector-transmitted diseases like malaria, in which Ava has worked, dengue disease, schistosomiasis, and many others that really are a burden for the human population. And there are others, another side, which are the environmental transmitted diseases like cholera, like Toxoplasma gondii, and many others. So I'm going to use this environmental transmission to illustrate the idea behind this linking between two temporal scales. The temporal scale at which the within host process occurs. Yes.
As you pointed out, some of us are sort of by now familiar with the picture on the right and not on the left. So vector means you have a little bug which flies from me to you. That's a vector. What does it mean, environmental?
Environmental means like you drink water or eat contaminated foods.
But how does my disease get into the water?
because I have
because I go to the bathroom.
exactly.
Yes.
Exactly. That is so. Of course, but this is a very interesting comment because many of the environmental diseases and some vector transmitted diseases are a product of economic inequalities and public health problems in populations, right? In Latin America, in Africa, in many places of the world. So that's one thing. So
if COVID gets transmitted through the air conditioning system in this building, would you call it? And so I don't infect you directly, but I sort of cough and it gets picked up with the air condition and gets pushed on you. Is that an environmental
no, well, it's
transmission?
considered an intermittent disease because it goes from an organism to another organism through the air, right?
It goes from organisms through the water to
the water, but we don't live in the water, right? We interact with this inert place in which they have eggs or certain life.
So I should imagine that it doesn't just go directly to the water, but then in the water it puts eggs or some, so that you need still some little bugs which do something in between to call it environmental.
You need several of these diseases have different stages, life history stages in the environment. But others like amoebiasis, for example, dysentery, are directly pollution, or cholera are directly the pollution of the waters in a large area in which you get sick when you drink, or salmonella and all these things. No? Then that's the thing. And now the immunological process, the model I'm going to be using is this one. This one is a very classical model developed, essentially exploited by Perelson and his group. And these are the target cells, the cells that are the target of the virus. Lambda is the production rate of these cells. K is the rate at which the virus penetrates that cell. M is the natural mortality for those target cells. D is the disease, the virus viral induced, the pathogen induced disease, death into the target cells that are infected. And then the other part, the important part here in terms of the viral production and the production of the yes?
T-star.
Sorry?
T-star is infected T cells.
You have T and T-star.
These are target cells, naive,
Yes.
and this T-star is infected target cells.
So it's
It's not a steady state or anything like that. It's just a
like
symbol.
SNI.
Yeah, it's a very
It's CD4 cells that are infected.
The difference is that now we are looking at the particle that produces the disease, and this P is the production rate. That is when per capita, so you have a cells, it explodes, suppose, and it liberates P virus per time. And this is the rate. And then
So far, no humans, right? This
is general for vertebrates. In general for vertebrates. So I'm going to speak about it could be inside human or inside practically any vertebrate. This is a very general model, but we can think on humans. That's okay. And C is the clearance rate. So the bioparticles live only certain time, 1/c is the time exponentially distributed that they live inside, and then they get out of the system. The model that I'm going to use in order to link these two processes is the SI model because it's simpler. We could put immunity here, but that is more complicated. So this is to illustrate what I want to illustrate. This is enough. This is just an SI model. It's a chronic disease. So you have the birth rate equal to the death rate, the effective contact rate. Those are the basic components. So we know many things about this thing, but the example that motivates us to work in this is joint work with Xilang Feng and other colleagues from Mexico, is Toxoplasma gondii. Toxoplasma gondii is an interesting disease because it practically all mammals in the earth are susceptible to Toxoplasma gondii, but the only ones in which the parasite completes the cycle, it can reproduce sexually and then produce oocytes that can infect, are the cats. So it's all species of cats are known to harbor Toxoplasma and to allow the reproduction of the parasite. There are problems with this. In Europe, there are even vaccines for pregnant women to protect against toxoplasma. Not here, not in Mexico, not in many places, but in Europe, yes. The sexual, as I said, is only on felines, I think is the pronunciation. And the point here is that the cat becomes infected, sheds all sites in the urine, in the feces, and then the environment is contaminated. The oocysts are deposited there, and they can last very long. Then it comes a cow, or it comes another mammal, eats and ingests the oocytes. All of us, except the cats, are dead ends for the toxoplasma, but it still can cause disease. That means that the toxoplasma cannot reproduce inside anything that is not a cat, but it can damage the body. So this is.
Eat the bird, so
Yeah.
if I eat the bird, do I get sick, or is it a dead end for the
No,
thing? So in a food chain, it can go many hops.
There are for example, there is an hypothesis, interesting hypothesis, that Toxoplasma gondii is a disease that has produced evolutionary changes. This is controversial in mice, because the Toxoplasma goes to the brain of the mice and then changes the behavior. And instead of being afraid of cats, they are attracted to them and they run about, and the cat bites them exactly in the head and gets infected. So you have a predation process there.
No, but if I now eat the mice,
Yeah, you can get infected. Yeah. So any mammal.
He goes to the restroom now, he eats mice.
So this. Yeah, because you got the cow, it's grass, and the grass can be infested with oocytes.
Sorry, because it's very interesting and scary. No, I don't want to be recorded.
Oh, she doesn't want to be recorded.
My cat has Toxoplasma gondii, and I played with this cat 10 hours.
You don't wash your hands, you can get.
Can you repeat the question?
So wash your hands after you.
Oh, yeah.
enter the litter.
Yeah, it's an environmental disease.
it's a litter. If you clean the litter,
You clean the litter, you clean your hands.
So now we have problems with cats, because not only do they give us gondii, they also gave us the first SARS, right? Civet cats were responsible for SARS 2002.
Yeah, so
Different cats, or the civet ones, yeah. These ones are domestic cats.
yeah, well,
I don't
Toxoplasma is very interesting, and it's a lot of things around, but this is the example. So you have a mammal that sheds viruses in the environment, and then from the environment, you can infect any other mammal, have the cycle. That's the reason. And the thing is that we have several times involved in this disease as in many others. The transmission times, which give rise to the epidemic, the infection times that give rise to the individual disease, and the environmental time in which the oocytes are resident in the environment, waiting to be ingested and produce disease. So this is the idea. And as in many other diseases, many, the size of the inoculum is very important, not only for environmental transmitted diseases, but in general, you can have it.
So in the SI model, do you have this contamination of the environment in the SI model, right?
I'll
later, okay.
I'll put together, because what I want to do is to link explicitly the immunological dynamics, the epidemiological dynamics, and I'm going to use that through
the
environmental compartment.
Nice.
So why do you call it SI and not SIS?
Because I'm not, because
you want to repeat the question?
Easier. I mean, Toxoplasma gondii, there is no, once you get infected, you can remain shedding viruses. Well, not you, general mammal, for long time. So that was our example. But in terms of the results I'm going to show, doesn't really matter if it's SIS or SI. So in terms of time scales, I'm going to use biological insight for this. So we have three times scales. The slow time scale, that I call it the epidemiological time scale, because it takes place in weeks, months, or years. The fast scale is immunological dynamics, the within host dynamics, it takes place in hours or days. And the intermediate scale, that just for calling it, is essentially a slow scale, that is the resident time of the oocytes in the environment. So this is the model for outside. I'm going to modify it. This is the model for the within host. I'm going to also change it. This is the equation for the environment. We have this logistic growth equation. It's an SIS kind of model. We have a carrying capacity. It's normalized to 1. And this theta B indicates the shedding of the virus per capita per infected cat or individual here, mammal. So you have, this is the contaminated environment that is going to link these two things. How is it going to do that? Well, before we go to that, I'm going to describe equilibrium situations. So just to remind you, this is the basic reproductive number for the disease. Very simple, lambda over mu times the number of susceptible population, the density of population, of susceptible population. And the other is this one. It's very simple. Lambda over m is the total number of available susceptible cells. K over C is the infection rate, the infection, actually the number of infected cells per capita that occur during the time in which you have viremia, which is before the virus is cleared. And P over mu plus M plus D is the net production rate during the time in which infected cells can be producing virus. So has a very, that's why we call it R naught also, but fancy R naught, and this is normal R naught. But these are the reproduction numbers that rule when you have an outbreak or an infection.
So humans do not get infected by contact with the contaminated environment?
Oh,
not yet. Oh, right. Sorry. Okay.
So this is the components. The questions I want to ask: how does the within-host dynamics influence transmission? What is the effect of disease transmission on the viral dynamics? And if we have time, some things that I could say on evolution of virulence when you couple these systems. Evolution of virulence is an interesting thing because it's a very classical area in these processes. In general, when you have a variants of a given virus or pathogen, the ones that are more common are those of intermediate virulence. The question is why? And one of the classical answers is that you have a trade-off between transmission and virulence. So if you are too virulent, you kill too many hosts, transmission lowers. If you are very mild, you will not get transmitted. Therefore, selection favors intermediate virulence. That is the classical result. So we want to know what happens when you link these two things, because you have two systems, each of them selecting for the best strategy in different contexts. So that's it. Now, this is the model. You have, since the population is constant, so I have omitted the susceptible population. N is the total population of the mammal. So if the infection of these susceptibles gets because they interact with the environment. The environment, I already described the equation. This is the theta V is the shedding rate. And this is the normal within-host mathematical model. But now we introduce this GOV, which is the inoculum, which has this form, just very general. This can be generalized. But in general, we have a linear dependence of the inoculum on the concentration of viruses or oocysts, in this case, in the environment. This is the linked system. We can define two times scales, the slow and fast, but we are going to, well, this is the shedding rate and this is the inoculum. This is the linkage between these two separated systems. And this is essentially the basic things I'm going to do, the approach I'm going to show you. So the between host dynamics is slow time scale. The within host is the fast. We are going to ignore the transient dynamics of the virus cell interaction because it's fast. So from the point of view, we are only going to focus on the steady state of the within host equations because we assume that they are so fast, they reach stationary state before the between host dynamics. And then we are going to feed that steady state dynamics into the between host dynamics to study the whole system. So by parts, right? So this is how we approach this 2 times scale problem. And first, we look at the isolated cases. So we have the isolated within host dynamics. That is, when there is no inoculation of viruses, you just have the disease, you stop the input from the environment. And this is the equations. This is the repetition of that. This is the basic reproductive number. I call it within R naught. So I save myself fancy letters. And T0 is going to be the total available susceptible number of cells, target cells in the absence of infection. So we have the normal result. If R is W0 is less than 1, the disease-free, infection-free equilibrium is global and synthetically stable. And if it is greater than 1, then there is a unique positive equilibrium inside.
Lambda M or what? No,
sorry.
T naught.
This should be M.
Yeah.
Right. Sorry. Yes, it should be M.
Fine.
Yeah. So, right. So this is the basic result here. And we want to look at the between host system. So we assume that these guys are in V is in equilibrium. So it's a constant that it depends on E, right? It's now E is a parameter because it's our steady state. And so this is the new within host reproductive number, now between host reproductive number. Look, this part is the normal R0, but now we have linked this R naught by with the within host dynamics. This is, as you see, this is the shedding rate during the time, and that can infect a reservoir, a mammal, during the time in which these oocytes are in the environment. So it's a very basic description, but okay. So we have a disease-free equilibrium, and the normal thing happens if RB0 is greater than 1, then you have an endemic state that depends on the reproduction number. But now this reproduction number is different. And so now we want to put together things and see how this interaction affects what we know about each system when it's isolated. So within host now, the interaction. This is the steady state for the within host system. It depends now on E as a parameter. So, and one important thing is that when you have the contaminated environment, that is that the G of E is not 0, then the virus-free equilibrium, the pathogen-free equilibrium does not exist. So you don't have a system in which you have 0 virus. No? When you have inoculation, you always have viruses inside you.
Sorry, I have a question.
Yes.
Do you have any restriction on what form G of E should take, the inoculum? Should it be like
G or function G is a linear function for now. But essentially, you can take it as
type 2.
E to the gamma, where gamma is less than 1, for example. Or well, C letter.
Well,
so
saturation function, maybe.
Yeah.
Whole in type 2. Okay.
But then now is linear. It's easier. Then I just put these expressions there because these are just formulas, but you can solve the differential equations for the equilibrium and you get this. And my only intention for doing this thing is to show 2 things. One, that now these expressions for the equilibrium values of infected cells and target cells, naive target cells, now depend on the within host reproduction number. And since these things are V now is affecting, is going to affect the dynamics of the between host system. So just to make sure that you have this picture there. Now, this thing that appears in the basic reproductive number for the between host system depends on RW0. So we have linked both systems. And one particular thing is that if RW0 is greater than 1, you can take the limit when the environment goes to 0, that is the contaminated environment disappears. So when this goes to 0, then if RW0 is less than or equal to 1, then the limit is 0. But if RW0 is greater than 1, then this is the viral load that you will have in that case.
So I
Yes.
have one more question. Sorry. So the reason the virus free equilibrium doesn't exist is what? Because GOV
Because
is not
GOV,
zero.
and
It's not
you
zero.
see the
Okay.
equations,
You have influx from the environment. Okay. It's not zero.
is not zero.
Okay.
So
Fair enough.
So I have a related question, which seems that for this to hold, you really want to think of it as that environment really acts really fast. And then it sort of serves almost as an external replenishing of the virus inside the population. If I made the times the other way around, would it be the other way around? Would then only they are not, the latter are not matter and the other one not?
well, you're right. Yes, I think I'm.
So does it mean that in reality, actually both do matter? Because,
Well, yeah,
because
yeah.
it's not infinitely faster, it's just 1,000 times faster.
Yeah. Well,
So would I need both R's to be really larger than one, or what is the?
well, we'll see what happens under this hypothesis. In this hypothesis, you can have R within the between host reproduction number less than 1, and you still can have equilibria. But you are right. Probably I should be clear on the dynamics of E, because why I'm assuming that E is at equilibrium, right?
Right. So you have already one of them in equilibrium, and then you use it as input for the other.
So I'm assuming that these guys, to the environment and the between host systems, are constant from the point of view of the fast system. So it's so fast that changes in the other subsystems are not noticed. So I assume those are constant.
Right.
That's
So if I change the order of which is fast and which is slow, would that change the order of the R naught, the importance of one R naught versus the other? Or as a,
but that could, but that wouldn't happen, right? Because you cannot have an epidemic faster than the infection it takes inside the body. Because you need to transmit. So the within host system has to be faster than the scale at which you are producing an outbreak. So it's a biological argument.
But in principle, you're right in the sense that if you have two different reservoirs and one of them is on the faster scale, then depending how you set it up, one of these R naughts will matter, but the other will not, right? So
but here
that's true. But here I think that's right.
I thought this is invasion of production, but because one is at steady state and you introduce another.
Yeah, okay.
Essentially, this is
It's an invasion of production.
I think so, yeah.
And we'll see now how they interact. But in this case, both play a role. Okay. So I haven't thought in the possibility. So now for the between host, within host, these are going. I'm going to what I said in a moment ago. This is the slow system, both of them. So this will occur slower than the dynamics at the individual level. So if this thing is greater than 1, then as I showed you before, the limit when the environmental contamination goes to 0 is this one. So this is the initial viral density. And we can build the new between host reproduction number that now depends on this parameter. Right? So I have now this thing linked. So RB naught, here I already described this. This is this and that. This should be M. This is the basic reproductive number of the isolated system. And this is the production of the shedding of viruses during the time in which the environment is present. And here we have that the disease-free, this is classical result, 0-0 infection-free equilibrium. If RW0 is greater than 1, W0 is asymptotically, locally asymptotically stable if the reproduction number between hosts is less than 1, and it is unstable if this is greater than 1, which is the classical result. If this is less than 1, then it's less. It's just locally asymptotically stable, the disease pre-equilibrium. Okay, so questions? Okay. Now, I already described that, so I skip it. Just to show you, these are simulations to see that one is fast than the other. This is the but this is the within host system. This is for the target cells, infected target cells and viruses, and this is for the environment, for example. And we have these two different scales. Obviously, it's faster. So just to illustrate that under reasonable parameter values, biological significance, this works as stated. And the point is that here can appear multiple endemic equilibria, and this is where both reproduction number play a role. So I put here the dynamical system we're looking at, and by solving these two things, we can have I and the expression for I in terms of E hat. This is the constant value of E. E is a solution of this function, which is a complicated function, but it's algebra simple, but we have several cases. If R W0, this is the within host reproduction number, is greater than 1, and the within host between host reproduction number, this is the epidemic reproduction number, is less than 1, then we can have several scenarios. H max, the maximum of this is a concave function. We have two possible endemic equilibrium that exist. I will talk about the stability later. If is 0, then it's unique, and this is less than 1, no equilibrium exists. If RW0 is less than 1, then these two exist, is unique, and no equilibria in the other case. And if both are positive, in this case, that's probably the thing they were thinking. In this case, this only one. It doesn't matter what the value of this, as long as they are greater than 1. And the only comment is that is H max requires that this derivative be greater than 0. That requires that the inoculum function satisfies certain, is bounded below by RW0. So everything is connected here. And the important thing is that you see the R naught of the epidemic system has two equilibria when RB is less than 1. That's the thing. So this is what I say here.
So it'll be two positive equilibria.
Two positive equilibria. So feasible, biologically reasonable equilibria.
And also the disease-free equilibrium exists, right?
Yeah, for the between hosts, yes.
Yeah.
For the other, it doesn't. Yes. So this is what I have said.
Now, given that the time scales are not. So given that the time sets are not infinitely related, you may wonder whether if sort of one is just on 1,000 times slower than the other, maybe one of these becomes unstable or not.
I'll see the next I think it's the next. Oh, well. A discretion of backward bifurcation, but this is what we are looking at here. So in general, in general models, you have a parameter, a reproduction number usually, and as we were discussing last 2 weeks and a moment ago, essentially when you have a transcritical bifurcation, this slope is positive. As you move the parameter above 1, then you lose stability of the disease-free equilibrium and you gain stability in the endemic equilibrium. And this branch is a continuous function of the parameter. Here, you don't have that scenario. You still have the transcritical, but now the slope is negative. So it's a different picture arises because now you have two endemic equilibria. One is unstable, the other is locally stable, and the disease-free equilibrium behaves normal. The problem here is that we have a problem of hysteresis, right? If we use R as a control parameter, and we are in a neighborhood of this equilibria, we'll be going, suppose we start here, we increase the parameter, instead of going up slowly, suddenly when we reach a certain value here below 1, we have a jump, and this will be the attractor of disease. And the same way, if you go backwards, then you'll jump to the disease equilibrium until you get to this part. So you get attracted, you jump here, you follow, you go the other way, you rise later. So you have a hysteresis phenomenon. Yes.
There seems to be a piece of a curve below the
Well, because I draw it. That's my lack of ability with Mathematica. I
Yeah.
couldn't get rid of that. Here, this is what's done by one of my students, and I tried to reproduce it here, but I couldn't. But no, actually, I think it's correct. If you don't care about biology, then this is stable because this actually goes like this. So this goes like this and below, but
It's
it
stable,
doesn't.
but it's
For us, it doesn't.
Negative infection rate. Yeah.
Right. So we don't care.
But his two models, this paper is extremely influential. It's one of the first few papers to establish the presence of backward verification in epidemiological.
Yeah, this is
Your two models, you have to claim the credit.
The model is very simple. You have mortality, you have a vaccination rate, the disease wanes, the vaccine wanes, and probably the natural immunity also is temporal. Then you have V. This is you can get from this vaccinated. Then the vaccine has certain efficacy that can protect. And these guys it wanes, people die. And then infected is just beta Si plus minus sigma beta Bi minus gamma plus mu I, and R equals gamma I minus mu plus theta R. This is the basic thing. So the reproduction number here is the normal R naught, which is beta over gamma plus mu, times, if I remember well, it has to be something like phi minus sigma phi plus omega plus theta divided by phi plus omega plus theta. So this is the basic reproductive number for this model. And in this model, essentially what you have, let's call it V, you have R0 and a function phi of certain parameters. In this case, the parameters would be phi, omega, and theta, and sigma. And depending on where are these parameters, you can have either this diagram or you can have that diagram. So essentially, if you have, in general, because I haven't looked at the space that these things generate, but essentially for low efficacy, low coverage, and short warning, you are more prone to have this scenario than this one.
Yeah, but if you have a perfect vaccine, for example, you have that. You have the transcritical, not the backward.
And then, we naively, because it was a naive exercise, we substituted the values that we have, or the different vaccines that we have for COVID. In Mexico, we applied 7, substituted into this very simple model, and all of them, fortunately, render this kind of thing. So, but this is the classical backward bifurcation, and this thing appears in the model that I'm describing to you. Okay, questions? Everything?
So what would be the mechanism that causes backward
Well,
verification?
in this case,
this one we know is the vaccine being imperfect.
Yeah.
But in the other one, what causes?
We'll see.
Yeah, what causes that will be interesting.
So why did you say luckily? What would be bad if COVID looked like that? So why would be bad if COVID
to eradicate the disease, you have to take the reproductive number way below 1. To reach 1 will not be enough, like in this here. So you will be, the little thing here is not that good. If I exaggerate the thing, this should be like this, and this is 1 table, and then that's a more visually clear picture. So that's the point. Here is the true parameter that will get rid of your disease very suddenly. It will be a jump.
So in this case, backward verification makes the effort to control disease a lot more difficult because it depends on initial conditions. One R not less than 1. If it's above the stable manifold of that saddle point, then you go up to the stable branch. If it's below, you come down to.
Yeah, I'll show some pictures.
Oh, sorry. Okay.
No. So we have that published some time ago, and a whole new field of things develop from here. So in the case, coming back to our to the model, this is essentially the function h, and you can see how we can have two positive equilibria for values lower than a certain, this is the threshold, the R star that I put here. So this is below 1, it's above 1, and this is the same picture, but with all of them below 1. So we can see that we can have both two equilibria, endemic equilibria in this case. Okay. And this is what Ava was describing. If you use the trajectories, then there is the stable, the unstable state is somewhere here. So it depends on initial conditions. You start with a large number of infected people, you will go to the endemic state. If not, you will go to the disease-free state. So this is essentially the thing. And now on the stability, it's described here, but you can look at the pictures. This is locally asymptotically stable. This is locally asymptotically stable for these conditions, right? And this is a saddle node, the one that is here. Okay. So that's the full picture of things. Are now, what produces this thing? that's one something, and it has to do with how willing. I don't know if I have it. Well, this is just the picture of the slow manifold and the fast manifold, right? How it goes. So it works for the within host system. Let me see if I have the pictures here. This is just, I mean, further description. You have a large region of multiple interior equilibria and so forth. Let me see if I have what you asked. It has to do with in this H, right? In this case, for the vaccine, what you have is, since the efficacy of the vaccine is not large enough, then you have a surplus of susceptible population that is fed into the general population that is affected by the disease very quickly. And so that generates probably that. And in this case, I think is the same, but now the environment plays that role, like holding for certain parameter values, the speed at which you are feedback into the endemic state, into the dynamics of the disease generates surplus.
We have a similar setup for cholera, but we didn't have a barcode verification. I think we have contribution from.
So you have this thing,
but it's not logistic. Yours is logistic. We had
and remember that this E depends on the within host. So it's this E depends. Where is this? Yeah, this V depends on RW0. So this also plays a role. So it depends what is the value of this in order to have the others. If this is greater than 1, then if you this number is no. Oh well, I just substituted sigma, phi, the coverage, the sigma, and the one for in this model, and I got the. On the list, this is an error.
No, but I want to, you simulated that for COVID.
Yeah.
So what is the COVID? What should I imagine? What is the environment? What is?
I don't know. I simulated this for COVID.
Oh, I see.
This for the other is other thing, right?
Yeah.
Well,
my guess will be, I think the mechanism leading to barcode verification in the model is the logistic contamination of the environment.
Yeah.
If it's not logistic, if it's linear, for example, you probably wouldn't have that. It's a quadratic non-linearity, probably. But
yeah, because IH is a quadratic, right? That's why you say that. Yeah.
Yeah. But it's very interesting to find what's the reason.
So,
very nice.
So that's the thing. And well, I'm going to be quick on the following. The next problem that we want to approach is the evolution of violence in general. systems, biological system, optimize certain parameters to improve fitness, right? The capacity of reproduction and be successful in an environment. The problem is that we have a system at the individual level in which the virus is going to optimize the way it invades the disease, the cells, and the other is the transmission to have another host in order to invade that host and generate the disease there. So in general, you have a trade-off between virulence and transmission, in this case, infection within host, and you have virulence and transmission in the between host system. And the question is, will the same value of the key parameters will coincide in both of them? In what I mean, well, let me skip this, it's just a description. We'll now incorporate disease-induced death rate in order to measure virulence better and have this trade-off clearly stated, because now this is the disease related to the production of virus. And this is the same model. But now, you remember in the reproductive number, P over M plus D, we call it phi of P. P is the production, the birth size, this whole thing, but P is the production of virus per cell. And now we are going to make the killing of the target cell, infected target cells depend on P, right? And this is the measure. This is the measure of fitness for the virus inside the body. And as an example, I'm going to assume that the shedding of the viruses has this form with C positive, that the disease-induced death rate depends on the target cells. This is the equilibrium target cells after infection. So this number is smaller than this one. And so this is a measure on how damaged is the tissue. And therefore, we assume it as an indicator of virulence. So the death rate is directly related to this difference. This is a way of doing it. There are several ways of doing it. And where D is, I've already said that. And so we are assuming also that biologically, the viral production rate is bounded, is a maximum, has a maximum. Biologically, it cannot grow forever. So you have a bound on the number of viruses that each cell can produce, right? And with these forms, then there is a critical point here for D. We have that, and we'll define P star as the minimum of P max or the optimum, right? So this is as far as the viral production rate is concerned. Then we can again solve our equilibria now in terms of phi, and we can look at theta, the shedding rate, as a function of P or a function of delta. Remember, delta is the disease-induced death rate, and P is the production rate. So we can look at them both ways. The same, this is the same. And now the reproduction number, the between host reproduction number, we can look it as a function of P, and we want to optimize this in terms of P, or we can look at it instead as a function of delta.
Sorry, is it P the burst size or number?
P is the
rate
at which virons are produced.
Oh,
and it is related to the burst
size. Okay.
Phi. Right.
Okay.
And then what is Z? I'm sorry.
C is a parameter that allows us to shape the form of theta. So we will C can be is positive, and I'm going to show you an example with Z less than 1. Then we have an optimal value. We can compute these things. I'm going to be quick on this. And let's call P plus and delta plus the optimal values derived from the optimization of these quantities for the between host dynamics for the case Z less than 1. And if you do, I think, just to conclude, pictures are more than 1,000 words, they say, and you have two scenarios, right? Essentially, if you disease-induced death rate is less than the optimum calculated before, then you have that the basic reproductive number for the within host system and the basic reproductive number for the between host systems are optimized for the same value of the viral production rate. But if the max, the optimal is less than the maximum disease-induced death rate, then the maxima are different. For the within host reproduction number is a larger number than that required to have an optimal transmission trade-off in the within host reproduction number. So you have this contradictory, well, it's not contradictory, these different aims that different elements of the system have in order to produce an infection. And I think that with that, let me just, we're looking at vector transmitted diseases too, but that is to prolong too much this. And this is what I have to say to you. Thank you very much. Questions?
Are there any questions, comments? That should be one question. I will still keep on asking you about the mechanism that I'm very interested in the mechanism that causes the microapplication, because it's the first time I'm seeing it. I've seen many coupled models that couple immunology and epidemiology, but I don't remember seeing any that has a backward application.
But it has to do with the inoculum rate, the G.
Yeah.
And as you say, the logistic growth of the environment, that you have a saturation in there. That could be, yeah.
So no, not to that particular question. I probably do not. But my question is different. So it seems like within host you're assuming certain the spiral like system for the interaction between cells and the virus. How much individual heterogeneity would affect this? Because right now you have these sort of averaged, idealized model, and there should be lots of variation among different hosts, right? Is this going to destroy potentially some of those effects?
I don't think it's going to destroy it, but you are right. This is an average behavior, and really what we should have is probably age of infection, right? You have the age of infection, you have a distribution, and then based on that, you can look at the same problem.
Or you could have some individualized behavior of these immune cells, or some kind of different infectivity within.
Right. Well, the thing that makes this work is that the within host system has a unique, globally asymptotically stable positive equilibrium. That allows us to take the steady state values and plug them into the between host system to derive all these things. Mm-hmm. If the system has cycles or the disease-free, the endemic, the equilibrium of the within host is not globally stable, then probably the same thing holds, but then you have to do much more work to prove that. This is based on that particular thing. And I underline this because there are many ways of modeling the interaction of the immune system with pathogens. And we chose a very classical one, special one. This is a question.
Thank you. So very much like Abba, I want to think about this backward bifurcation. Could you put the equations up that into that? That's
not this one.
Not this one.
The one that you see.
Yeah.
Okay. Yes.
Okay.
I can get rid of the circles. Yeah.
Okay.
Without the E, that's no longer the E plus contamination.
The contamination and the way it's
the I times B. So somehow it's got to be that IB.
This is a logistic more kind of
IB term.
The quadratic term, yeah.
Having more I around.
This is the shedding. This theta B is the shedding. So
it's essentially triple mass action, right? ID times E.
Yeah, ID times 1 minus E.
1 minus E, yeah. So
the rate of increase depends on the virus.
And then E shows up in the G of E term.
Then
going
when
into
you solve for
equilibria.
equilibria, you substitute
And it also shows up
in the expressions.
So
So
which one do you solve first?
for the fast system, we consider that this is constant because this goes fast. So from the point of view of the dynamics of this system, these are constant values. So we substitute.
T system.
Yeah,
and then we solve the coupled V, E, and I system.
E and I system, right?
So that's why I would want to answer the question mathematically is to apply the center manifold reduction. To do the center manifold reduction to determine.
We did that.
Oh, okay. So from that inequality, we'll know exactly what.
It's complicated. It's a lot of.
Yeah. No, that's very nice.
It's a confusing thing in the terms of algebra, but we did that.
Okay.
In the paper is a formal thing on singular perturbation with the epsilons from here from the equation, things like that. So the paper is it has everything there.
Oh, excellent. Okay.
Well, not everything.
Well, most of it, yeah. So the other thing I will ask you also relates to. Okay, so in this case, the production rate of uninfected CD4 cells is constant, right? So, but for HIV/AIDS, for example, we know during the AIDS stage, the production rate.
Which you mean P?
No, the lambda.
Here.
No. The T equation, the production rate.
Oh, this one.
Yeah. So at the end, I mean, during the AIDS stage, for example, the CD4 count goes.
Remember that this is for a general mammal, general system. It's theoretical.
Just keep it simple,
But if we specialize, certainly cases.
Yeah. No, that's really very nice presentation. Any more questions? Any more comments? Okay, in which case, let's thank Jorge one more time.
Thank you very much.
Yeah, sure. It's, I think the last, actually, no, the next talk is by Amin. I mean, you're given the last.
Tomorrow morning.
Oh, okay. I wanted to say.